Hello and welcome back to the Biochemistry for Health Sciences channel. In the previous videos, we talked about regulation of metabolism after a meal or the postprandial state and the role of insulin. In today's video, we shall start discussing regulation of metabolism during starvation. And today we'll talk about glucagon secretion. Starvation is a complex physiological state that involves several hormones. Here are a few examples. The hormone ghrelin increases to promote hunger. In the liver, glucagon promotes breakdown of glycogen or glycogenolysis, synthesis of glucose or gluconeogenesis, as well as synthesis of ketone bodies or ketogenesis. Cortisol stimulates gluconeogenesis in the liver, while the catecholamines such as your epinephrine and norepinephrine will promote glycogen breakdown or glycogenolysis in the liver. Cortisol, growth hormone, as well as the catecholamines will promote the breakdown of lipids, especially fat, lipolysis in the adipocytes, while leptin, the hormone leptin, will decrease to promote hunger as well as conserve energy. In the muscle cell, the catecholamines will stimulate glycogen breakdown, glycogenolysis, while cortisol will stimulate the breakdown of muscle proteins or proteolysis. During starvation, the thyroid hormones will decrease in order to decrease metabolic rate. In today's video, we shall focus on glucagon. This is a hormone produced by the alpha cells of the pancreas. As you remember, insulin is a hormone that also comes from the pancreas, and that comes from the beta cells of the pancreas. The alpha cells will first make a protein called proglucagon. This protein will then be broken down into several other proteins, what we call as proglucagon-derived peptides or PGDPs. And examples of these PGDPs are GRPP, which is glycantine-related pancreatic polypeptide, glucagon, intervening peptide 1, as well as the protein major proglucagon fragment. The enzyme that converts proglucagons into PGDPs belong to the family of enzymes called prohormone convertase family, and an example of this enzyme is PC2. So while glucagon comes from the alpha cells of the pancreas, it's important to note that glucagon-like chemicals are also produced in the intestine by the L cells and perhaps even in the brain. In the L cells of the intestine, proglucagon is broken down by the prohormone convertase 1,3 or PC1,3 into several other types of proteins, including glycantine, glucagon-like peptide 1, intervening peptide 2, glucagon-like peptide 2, GRPP, which is your glycantine-related pancreatic polypeptide, auxin to modeling, as well as glucagon 1 to 61. These peptides such as GLP-1, GLP-2, glycantine, as well as auxin to modeling are involved in appetite, regulation of bone resorption, glucose regulation, as well as in the gastrointestinal growth. Research on glucagon physiology as well as glucagon pathophysiology has been very challenging. What are the reasons for this? Let's look at why studying glucagon has been a very difficult thing to do. First of all, glucagon is very difficult to assay, to analyze. One of the best ways and the quickest ways to analyze or to measure the amount of a protein is a technique called immunoassay. For immunoassays, we require a good antibody that will react specifically with that protein and no other proteins. However, the antibodies that are available for glucagon unfortunately may also react not only with glucagon but with other types of chemicals that resemble glucagon, such as glucagon 161, glycanthin, proglucagon, as well as oxyntomodulin. Researchers have used several different types of glucagon antibodies. One that reacts more with the N-terminal part of glucagon has been used in several research papers. Unfortunately, this is not very specific. Similarly, there's an antibody that reacts with the site of the glucagon molecule. That too has been found to be not very specific. Probably the best glucagon antibodies available so far is the one that reacts with the C-terminal end of glucagon, the C-terminal antibody. This is much more specific compared to the other antibodies. However, it still reacts with this glucagon-like substance called glucagon 1-61. Besides non-specific antibodies, there's another problem is that several of these glucagon-like chemicals are also secreted. For example, proglucagon, glycanthin, oxyntomodulin, as well as glucagon 1-61. And when these chemicals break down, they can give you breakdown products that resemble glucagon very well. And because of this degradation of circulating glucagon-like molecules, we might get a falsely increased glucagon when we measure the glucagon by immunoassays using antibodies. Another reason why studying glucagon physiology as well as glucagon pathophysiology has been very challenging is because of the complex regulation of glucagon secretion. Glucagon secretion by the alpha cells of the pancreas is affected by several factors. One is the paracrine effect, we'll talk about. The other is the effect of nutrients on glucagon secretion by the alpha cells of the pancreas, as well as the effect of other hormones on the secretion of the hormone glucagon by alpha cells. So let's look at a little bit more about these effects. Besides the alpha cells in the pancreas, as well as the beta cells that secrete insulin, the alpha cells secrete glucagon, there are other cells found in the pancreas that secrete hormones. So one of them is the delta cells. So when glucose concentration increases approximately more than 130 milligrams per deciliter, that's about 7 millimolar, that causes increased secretion of the hormone somatostatin by the delta cells. Now somatostatin, so this hormone somatostatin, will bind SSTR receptors, somatostatin subtype receptors, on alpha cells. And when this somatostatin binds this receptor on alpha cells, glucagon secretion by alpha cells is strongly inhibited. In another paracrine regulation, insulin that is secreted by beta cells, so here's insulin secreted by beta cells. When this insulin binds the insulin receptors, which are also found on alpha cells, so when insulin binds insulin receptors, glucagon secretion once again is inhibited. Glucagon secretion by the alpha cells is inhibited. So somatostatin as well as insulin affects the secretion of glucagon by alpha cells. Some research papers have also shown that there are several other proteins, several other peptides, such as one example is urocortin 3. And when urocortin 3 or peptides like this is secreted by the beta cells, this peptide will go and bind to the delta cells and stimulate the production of somatostatin. So somatostatin rises, and that will go and bind to the SSTR receptors on the alpha cells, and the somatostatin will, as we mentioned earlier, inhibit glucagon secretion. So indirectly, peptides coming from the beta cells might be decreasing the secretion of glucagon by the alpha cells. Another paracrine effect is carried out by glucagon itself. Glucagon secreted by the alpha cells will bind glucagon receptor, so glucagon receptor present on beta cells, as well as GLP-1 receptor. So here's a GLP-1 receptor. So glucagon will bind these receptors on beta cells, and that will lead to increased insulin secretion by the beta cells. And as we discussed earlier, insulin will then come and bind to the insulin receptors on the alpha cells and decrease glucagon secretion. So clearly there is a very complex interrelationship between the alpha cells that secrete glucagon in the pancreas with the neighboring beta cells as well as the delta cells. So besides the paracrine effect, another factor that affects the secretion of glucagon by the alpha cells are nutrients. For example, hypoglycemia, when your blood glucose concentration is low, that will stimulate secretion of glucagon by the alpha cells, while hyperglycemia, when your blood glucose is high, will inhibit secretion of glucagon by the alpha cells. The mechanism of how hypoglycemia, low blood glucose, causes increased secretion of glucagon, increased exocytosis and secretion of glucagon. This mechanism is not very clear, but it resembles that we discussed in the secretion of insulin by beta cells. And that includes the production of ATP from glucose, which then will affect these potassium ATP channels, lead to depolarization, and that change in the membrane potential will be sensed by voltage-gated calcium channels, which will open, and once they open, calcium levels inside the alpha cells will increase. Calcium will enter the cytoplasm, and the increased calcium will lead to increased exocytosis and subsequent secretion of glucagon into the bloodstream. So as mentioned earlier, is exactly how this is happening. The mechanism is not clear. Some researchers believe that the effect of glucose on alpha cells may not be directly on the alpha cells, but may occur through paracrine regulation that we discussed earlier. Other nutrients, especially fatty acids and amino acids, have been suggested to be more important for regulating glucagon secretion than glucose. Amino acids, especially arginine and alanine, stimulate glucagon secretion. They're very strong stimulators of glucagon secretion, while fatty acids typically inhibit glucagon secretion. So besides the paracrine effect as well as nutrients, Another reason why studying glucagon physiology and pathophysiology is challenging has been the effect of several other hormones on glucagon secretion by the alpha cells. So, for example, the catecholamines that may come from the nerves or the adrenal glands, they stimulate glucagon secretion. So while the catecholamines stimulate glucagon secretion, there are several GI peptides that come from the gastrointestinal tract, such as your glucose-dependent insulinotropic peptide or GIP, GLP-2, oxyntomodulin. All of these stimulate glucagon secretion, while the peptide, glucagon-like peptide or GLP-1, inhibits glucagon secretion. So several hormones affect the alpha cells and subsequently the secretion of glucagon by the alpha cells. So besides unreliable assays for glucagon because of the non-specificity of the antibody used against glucagon, the complex regulation of glucagon secretion, there is also species differences in studying glucagon. For example, glucagon is well known to stimulate lipolysis in rats, but we don't see that in humans. Another problem with glucagon is it has a very short half-life. It doesn't stay around in the blood too long. So once it's secreted, does its function, it is then excreted or metabolized. So it doesn't stay around too long for us to analyze it. Dose-dependent effects. So when researchers give low doses of glucagon, they see certain effects. When they give higher doses of glucagon, they see different effects. And that might be due to the interaction of glucagon with other receptors, such as GLP-1 receptors. And then we could have direct versus indirect actions that are mediated by the sympathetic, parasympathetic nervous system that release catecholamines, and that too can affect the secretion of glucagon by the alpha cells. In the next video, we shall look at the effects of glucagon on the human body, keeping in mind that due to all these reasons, these effects that we're going to discuss in the next video has also been an area that has been very challenging. So until the next video, thank you very much once again for watching and supporting this channel. Goodbye and have a wonderful day, and hopefully we'll see you soon in the next video.